My research in the past few years has also been particularly about this topic of breeding for intercropping, or how do we breed for diversification. So, but this morning is sort of an easy introductory lecture to the topic. How do we go about it? Now, I don't need to motivate shall we say, the why we need to think about intercropping and intercrop breeding. You've probably seen this type of picture already, how humanity is essentially transgressing these planetary boundaries. We're really pushing past the safe space that we've been operating in, or this planet has been operating in for millennia. And one of the major drivers of a lot of these transgressions is, of course, agriculture, particularly agriculture geared at producing animal-based proteins. I mean, we could talk about that, but agriculture as a whole, the whole agricultural system we have is doing a lot of damage, as we know, to nutrient cycles, to water cycles, not least to greenhouse gas emissions and so on. So we clearly need a transition in how we design our agricultural systems. And this is why I presume all of you are here and all of you are interested in this topic. And probably you've already seen this slide or this figure, how diversification strategies, be it agroforestry, intercropping, crop rotations, but also mixtures of varieties, how they can have positive impacts, but that these impacts are variable. So if we look at these production type services that we can get, we see indeed that in general that we can increase productivity if we have a more diverse system, but that is not always the case. And what we thought about, at least when we were approached originally by Jochem Evers at the plant breeding group, he was one of the people that approached plant breeding and said, well, what are you doing about intercropping? And one of the first questions we thought about is the modern varieties that, at least at the plant breeding group, we seem to be so focused on, these elite varieties, are these adapted for systems that are no longer monocultural? And if they're not, how should we breed for these systems? So this is a sort of the basis for what I'm going to be talking about this morning. Now, a number of us, including Jochem, if you can just see his name, and others from Wageningen wrote a review about this a few years ago, about breeding beyond monoculture. But a couple of months later, Virginia Moore and colleagues from Cornell also wrote a review about the same topic, breeding for intercropping. And one of the nice things about Virginia's paper is that she had this very colorful and descriptive decision tree, or a decision wheel, which sort of gives you yes, no type flow chart. So you pose a question, you answer yes or no, and then you move on to the next. So this sort of gave people, or at least it had a nice summarizing way of defining the problem space and what sort of actions we should be thinking about, depending on the outcomes of these answers. Of course, I'm not 100% in agreement with all of these, so I will also take a little bit of a critical look at some of these, but in general, it's quite a nice introductory framework for this topic. The first place that I'm going to start on is what Virginia talks about, defining the solution space. So can we solve the problems, shall we say, in these intercropping systems? Can we solve it more easily by, for instance, a management intervention or some sort of engineering solution? And it's, again, it says yes, no, and if the answer is yes, so if management, for instance, can be more effectively used than, for instance, genetic changes, well, then the sort of the quick answer is don't bother with breeding. But in a sense, this is a little bit, as the Dutch would say, kort door de bocht, so a little bit short through the corner, in the sense, perhaps it's non-trivial, and particularly if you have interactions between genotypes and management interventions, for instance, plant density, which we know often interacts with genotypes, or fertility, then it's not so easy just to say, well, you know, ignore the genetics, we'll just focus on the management. So it could be a little bit more nuanced than that. The second point is regarding do we need to breed for it, is to assess variation in the system. So do we have variation for performance in these intercropping systems? And again, the answer is, if it's no, well, then don't bother with breeding. But I would argue, if the answer is no, well, maybe you haven't looked far enough into the genetic diversity. Perhaps you need to test wider germplasm. Maybe you're too focused on elite cultivars. Maybe you need to move further afield into well, less modern germplasm, shall we say, land races, gene bank accessions, and so on. Or you might need to introduce novel variation. Maybe you also need to think further in terms of the crop species you're testing. So again, it's maybe not so simple as just to say, well, we don't see a variation, therefore don't bother with breeding. Maybe you didn't test sufficient variation in the first place. Okay, if we move to this green quadrant, and this is about designing the breeding schemes. Again, we have a number of questions posed by the authors here. And the first is regarding testing your crops in monoculture and in intercropping. So it's fairly simple. We have varieties of, I don't know, sugar beet, wheat, maize, whatever the crop is, and we can trial them in our monoculture system. We can also trial them in an intercrop. And if we pick up a significant genotype times management interaction, then it's sort of, according to these authors, the one of the measures for whether we should pursue a breeding strategy or not. And that has effects on whether you progress, for instance, to maintaining a monoculture selection program. But I'm going to motivate a little bit later that maybe G times M is not the best criterion for making this decision. So we can have a little look about that and how actually we could maybe have a better criterion for making this decision about whether monoculture selection schemes are appropriate. Okay. The following point is identify traits of interest. So supposing we have a G times M interaction, can we observe our key traits in monoculture? So again, is this it's not a particularly easy point to answer because we may not know a priori what the key traits are. So it may not just be simple for us to say, well, let's screen for key traits and check them if they're observable in monoculture if we don't actually know what the key traits for intercropping are. And this is non-trivial as well if traits change between systems. So what about plasticity? And that's going to be something that I presume you've been talking a little bit about plasticity this week, no?
Not at all.
Okay. Curious?
Oh, I know what it is.
Yeah, no, I know. I guess this is when I step in here, I don't know what you have already heard this week, so I don't know, is this connecting nicely with what you've already heard, but
No, it's complementary. It's totally different.
Okay.
It's fine.
Okay. Well, we'll talk a little bit about plasticity then. And so that is how traits change between how, in this case, between different growing systems. So yeah, our key traits easy. I would argue that they're not necessarily obvious. And then finally, well, if we're not able to observe these mysterious and magical key traits in a monoculture, well, then we need to do some testing. And again, the testing procedures I'll talk a little bit about. It's, again, not so obvious. How should we design such testing schemes? And of course, this comes at a cost. If we want to set up experiments where we start testing, looking for complementarity between species, but also between genotypes and varieties, then we really start to get into big numbers. And then the final quadrant here, I'm going to focus on this in the second half of my presentation this morning. So I'm going to be talking for the next 40 minutes, and then there's about 10 minutes at the end for questions. Is that okay? Yeah.
Sure.
Okay. So let's start off with assessment of variation. So, I mean, the sort of the basis for breeding is genetic variation. It forms the basis of selection. If we don't have variation in the species that we're working with, we can't make any genetic progress. That's fairly, I would hope, fairly obvious. And of course, if breeders, and particularly if you're taking on a new breeding goal, in this case for intercrop performance, it's important for us to understand how much variation exists for that. So is it worth it or not? But I put a little question here. How do we define intercrop performance? Is that something that someone can save me from trying to define? Would one of you like to try to define what intercrop performance is? Or how a breeder would consider intercrop performance? More yield of the whole, or of, yeah. So maximizing total system performance on yield.
Yeah, or how much the roots can complement each other.
Okay. Yeah, these could be very, indeed, this, I would say certainly at a very basic level, system total yield of the system. So crop A, crop B, and maybe crop C, if there's more than two, would be a good indicator of performance. Maybe it also has to do with resource use efficiency, so how much inputs you put in for that yield. Yeah, okay. And that is already more complicated than a breeder normally has, because a breeder normally has a single species to work with. If there's more than one, then that total yield then becomes a little bit more complex. And we'll come back to that again in a bit. So this variation, this genetic variation, if we can get a handle on it, we would also like to know, is this variation different from what we would say standalone performance? So the normal way of us that breeders have of assessing genetic variation in monocultures, is this variation different between systems? And we don't necessarily know that. We'd like to develop a framework to estimate these components. Of course, it's in the past, breeders, or shall we say even currently, breeders are focused on trying to define the genetic merit of candidates of selection, so of individuals, accessions, lines that they're screening. And normally speaking, they try to estimate essentially a breeding merit, a breeding value, or some way of ascribing some sort, like to do with the selection index, some way of ascribing a value to that particular genotype. Now, when you start putting plants together, particularly across different species, and if these interact with each other, so suppose we're growing here two plants next to each other, and they have an influence on the phenotype, so an influence on how they ultimately grow. So for instance, you might have that the presence of this plant has caused proliferation and root branching in the first species, and also there's been maybe some shade avoidance response in this species, so it sensed this neighbor and it says, well, I better put on more growth up high, so it's increased its height. Part of the phenotype now of this individual here is expressed, in this case, in more leaf growth in this partner, and vice versa. Part of the phenotype of this individual is now expressed in how its neighbor grows. So there's this interaction between these two species. And there's various terms for describing these effects, particularly in the literature. Traditionally, they've been either called social effects, but here we're going to just use the other term that's generally used, indirect genetic effects. So this is the heritable component of a change in the phenotype as a response to a neighbor plant. Now, we could try to define these things in monocultures, but I don't want to get very complex into the topic of indirect genetic effects. Let's just imagine that we're defining here an indirect genetic effect between species A and species B, which is what's relevant for intercropping. And of course, there are components that we could ultimately like to understand in both of these. So there's, in green here, we have, shall we say, some direct genetic effect that a breeder would like to understand. So this is, again, the genetic component of the phenotype. Some of this phenotype is caused by environmental effects, but primarily we're interested as a breeder in understanding the genetic component of that. But part of this is also expressed in this case, biomass proliferation in the second species. So part of this individual's phenotype is now measured as a difference between, shall we say, the ordinary base phenotype and the response to this individual, and vice versa. And those components don't have to be the same. And they can, of course, be to do with different traits. But here we're just talking about maybe biomass. But it extends to any trait that you want, that a breeder would be interested in. And as a breeder now, the objective is no longer to maximize this direct genetic effect, but we also have to take into account the influence on the neighbor. So we'd like to, in crop one, maximize, presuming that these things are things that we want to increase, for instance, biomass or yield. But then we would like to maximize in crop one, this direct and indirect genetic effect, and also in crop two, the same. And these, we can call these general mixing abilities for both of those two species. But they're measured in different crops. And this comes back to this point about system performance. These crops may have different benefits, they may have different economic values and so on. So a breeder now has a slightly more complex selection target than simply just the yield of crop one, because these things are intertwined. Now, before we get any further on how we would actually define a selection scheme or for these components and maximizing performance of the system, it would be interesting to know, well, these indirect genetic effects, I mean, they look very extreme in this little example here. massive increase in shoot growth and also root proliferation. It looks very interesting from this little figure, but in practice, how large are these variance components? How large are these indirect genetic effects? Well, there's been a number of studies that have looked at this question. Benedict is here talking after me, so you'll probably see this picture again. This is his work, so I'm stealing his slide. But here, if we look at the indirect genetic effects for this pea and barley intercropping system, that's in this case for the barley fraction yield, how much of the variation in the barley yield was caused by the genotype of the pea? You can see it's quite a large proportion of barley yield, in this case, in terms of variation, was caused by the pea genotype, and vice versa. There's also a non-trivial amount of variation in pea yield caused by the identity of the barley genotype. If we look at a study in Arabidopsis, which was published a few years ago, for biomass, again, they found significant indirect genetic effects of the order of, shall we say, about 10% of the direct genetic effects, slightly more. So small, but certainly non-trivial. Okay, so that's the first taste about these direct and indirect genetic effects. This question about do we have sufficient genetic variation in modern varieties, and this is not really an easy question to answer. There's been an awful lot of improvement in varieties to fit, shall we say, modern, highly mechanized agriculture. And a clear example of that is what's happened since the Green Revolution. So when you got all these resistance genes being bred into crops like wheat and rice, but at the same time, also dwarfism was bred into these, and it had a huge boost in terms of productivity for these staple crops, because a lot of the assimilates were now being put into seed yield rather than into growing lots of straw. But it may be that, for instance, in wheat, there was good reason why wheat varieties were tall in the past. People used the straw for thatching. They were able to grow above the weeds and so on. So it may be that, again, modern varieties, although they're extremely high yielding, may not be the most suited if we want, I'll come to your question in a sec, if we want additional traits that are beyond now the scope of, shall we say, the portfolio of modern varieties. Yeah, now, right, so I'm moving on to this idea of G times M. So again, oftentimes I show this slide in a sort of a basic plant breeding course that I'm involved in about G times M interaction, and usually we're talking about genotype times environment interaction. And the basic idea of interaction, I presume that you understand a little bit about statistical interactions. Well, in this case, if we're trialing genotypes across different management systems, different cropping systems, and we see that the relative performance of these genotypes is not stable. So here we would have a stable relative performance. Genotype 1 in red is always better than genotype 2, and by the same amount on this y-axis. But if that relative difference starts to change across systems, then there's a G times M interaction. And a number of years ago, there was this paper that was published that looked across a number of cereal legume intercrop studies, and particularly focused on the interaction effect. So whether there was an interaction between genotype and cropping system, and in about 3/4 of the cases where there was an interaction effect reported, this was found to be significant. So you could say already from this study, well, in 3/4 of the papers where they have tried to look for G times M, they find it. So therefore, there is evidence that, we shouldn't be just looking at monocultures to predict intercropping performance. But if we dig a little bit deeper into it, there are usually two types of interactions that are talked about, scale type and rank type. Could you guess, or have you heard these terms? Which of these four would be a scale type or rank type?
It's a divergence and the convergence are scale type because the effect size changes, but the direction of the effect remains the same. But crossover would be rank type because there is a rank change. So under some conditions, system 1 is better than system 2, and in other conditions, it's the other way around.
Yes, very good. So these are the scale type interactions on this diagonal, so convergence and divergence. And breeders are usually much more worried about these rank type interactions because ranking, which is what a breeder essentially wants to do, they want to say, well, I've got 100, 200 lines here, and I want to be able to rank them from very good to not so good. If these rank type interactions happen across systems, across environments, but in this case, across management cropping systems, well, then it means that they're not able to predict performance across if they're just using their monoculture plots for ultimately for an intercrop. So indeed, there is an important difference here. And I ran a little simulation to try to show this, but I was told that this doesn't clarify it at all. But for me, it clarified it quite nicely. To try to explain a little bit what's going on here. What I did here was I simulated, oh, I should go back a little bit. I simulated very simply a scale type G times M interaction. So I simulate, this is the size of the ratio between the genetic variances between systems. So I essentially said in system 1, we've got a fairly tight genetic variance, and in system 2, that just blows up. So you get this big divergent interaction effect up to a ratio of 2. So, so this is scaling from equal scales, equal variances to, well, the variances are now at a variance in environment 2 is twice as much as environment 1. So very much like spreading out like a loudspeaker. What you see here, of course, if you run a linear model and you have a genotype in your model, the management, and also the interaction between those terms, some sort of an ANOVA model, and you look at the significance of that interaction term, it blows up. So this is the -10 or minus log space 10 of the p-value. So essentially just becoming an enormously significant interaction effect. So huge scale type interaction here being demonstrated. But what I also did was look at what's called the selection efficiency between systems here. And this remained perfectly stable across this range of conditions. Essentially, 95% is almost perfect selection efficiency. And this was a small simulation with 100 genotypes, a particular selection intensity of 0.33, and a correlation of 1 between systems. And I'm going to come back to these a little bit more. So what a selection efficiency here, it's a way of describing how well we select the best candidates in our monoculture when we're going to grow them in intercropping. And we just do that by comparing the number of selected individuals in the intercrop, and we compare that to the number that were also chosen in the monoculture. So the sort of the overlap. And we compare this against the baseline of how many would be expected by chance if we're just closing our eyes and picking out of the two systems random individuals. There's always an overlap, particularly if your selection efficiency is small. So if you're selecting 90% of individuals, well, then you take 90% of this group and they're the same group. And here, yeah, there's going to be a large overlap. So we compare it against sort of a baseline, how many would overlap by chance. And this is a way of us, again, to quantify, to motivate whether selection is effective in monoculture if the target environment is the intercrop. Okay, and what does that look like in practice? If we looked at this, here's 30 genotypes on the y-axis. Maybe it's their yield, I don't know. And the candidates that we select in both systems are colored green. So here's an example of a negative selection efficiency. You might as well have put on a blindfold and just pick names out of a hat, because here you're doing worse than you would expect by chance. This is a negative selection efficiency. Here's a very low selection efficiency. Again, you can see massive rank type interaction, also a little bit of maybe scale type interaction. But again, the candidates that are selected here in the monoculture, in this case, are really not popping up in the top third of individuals that are selected. And so on, you can see as we increase, the selection efficiency increases, those lines start to become more parallel. But even at 100% selection efficiency, so the top third of individuals that were selected in monoculture for their performance, and also they also coincide precisely now with the top third of individuals in the intercrop, but you can also still see some rank changes happening here. But that doesn't really matter. In this type of scenario, we just want to see whether the top third and the top third coincide. So the exact rank, the ranking here, maybe in terms of a recurrent selection cycle, maybe it doesn't really matter as long as they get through that selection decision. But the fact that we maybe didn't get the 1, 2, 3 or 1, 3, 2 maybe that's less important. Anyway, but just to say, even if we have rank type interactions, the selection efficiency, I think, captures for a breeder more information than G times M. And what we know about selection efficiencies in practice, well, unfortunately, there's far less published on that. So this was a paper that was published already a long time ago in Euphytica. But here you can see in a number of, if I'm not mistaken, maize with bean experiments, the selection efficiencies were all less than 100%. And yeah, in 1978, again, they should have been wearing a blindfold because they did worse than they would have by chance in that particular experiment. Okay.
I'm sorry.
Yeah.
The definition of the selection.
You want to see the slide? You will have these slides afterwards, by the way. One words, yeah, essentially it's the deviate. What you can imagine it in the sense of it's a correlation between the number that we select in this again in monoculture and those that we select in the number of individuals. So an individual, again, I don't consider it a single plant, but I consider it a candidate for selection, like a plot or a bag of seeds, a genotype, some line that we're collecting. So if a selection efficiency is 100%, it means that the individuals that we select in the monoculture exactly coincide with those same individuals would have been selected in the intercrop. So our correlation between, so the information, shall we say, that's shared between the systems is almost perfect. We can make selection decisions in monoculture that can be translated perfectly to the intercrop decision that we would have made if we trialed it. So basically means we don't need to go to the trouble of setting up an intercrop trial if our selection efficiency is very high. And as I said, for me, this is a far better motivator of whether we need to breed in monoculture or intercropping than G times M. That's a bit what I was trying to motivate. Is that clear? Bobke, promise me that you'll be asked on this formula after the course as well, in the test. Joking.
No,
sorry. No. Okay, so the next is these traits of interest, and it's very nice that Benedict just entered the room. Hi, Benedict. So breeders usually, so this is the topic of key traits. So, and I think actually this morning's session with Sabine as well is about traits. So this, I know far less about this topic than my subsequent speakers. But breeders often, they know the traits of interest. In fact, if a breeder doesn't really know what traits he should be breeding for, then he's a little bit lost. He really needs to know what are the key traits. So it takes a breeder many years to learn the market, learn what are the key resistances, what do growers actually want. But in intercropping, there may be other traits that may also be important, traits that a breeder doesn't necessarily know about, traits that may not be obvious from the outset. And this is, yeah, we're going to have some examples of this in a moment, but there could be two approaches a breeder could take. The first is what I would call an agnostic approach. So you believe that there's a trait out there that's important for intercropping, but you don't know what it is, a bit like agnosticism. So you record loads of traits, and then you start sifting through them to see, do some of those traits correlate with intercropping performance? And that's, of course, could be very possible nowadays if we've got all these high throughput platforms, we've got drones, we've got risotrons, we've got many ways of gathering enormous amounts of information about the trait space of plants. So this is certainly, I would say, technologically feasible. The alternative approach is that, you could use, shall we say, some prior information that you have. And this could be looking at other fields, for instance, to ecological theory, but how to design these agroecosystems, or we could use predictions from these from plant growth models, these functional structural plant models. And I presume you've also had some speakers on these models.
We didn't talk about it
okay.
this time, but there's a recording.
Okay, yes. Well, there was a paper from the group with that Wopke is working in a few years ago that looked at maize and wheat intercropped, and they tried to use these functional structural plant models to understand why was the intercropping of maize and wheat performing better than the respective monocultures? Why did it have an LEOR greater than 1 and so on? And it turned out, by running these plant models, they actually found that this yield advantage in the wheat, could be explained by more reproductive tillering, so increased tillering in the wheat intercropping in comparison to the monos, and also larger leaves in the border rows. So basically the plasticity in the wheat for, in this case, above ground traits was very important for yield advantage. So this potentially could give you some insights, or could give breeders insights if they run these sorts of plant models of what traits should they be looking for in order to make progress. And yeah, we can think about that if we have time afterwards, how you would implement that in practice. The alternative is this agnostic approach, or somewhat agnostic approach. I call it a regression approach. Again, I see Benedict at the back, and this is directly from Benedict's work. So you record a number of putative key traits, traits that you think might have some sort of association with intercrop performance, and then test whether these traits are associated with your mixing ability or with these direct or indirect genetic effects. And here's an example from Benedict's work. Again, I think he'll talk about this in a while. Yeah. So I can skip over very briefly, but they found in their study a correlation, in this case, genetic, or a correlation between the stipule length, which is the length of this flag leaf on the pea plants, and these general mixing abilities in these mixtures. But I'm going to skip over that because I don't want to. I'm also running a little bit out of time. Okay, testing schemes. How do we design such testing schemes? Basically, the search space becomes very large when we try to estimate compatibility between species. So there are studies, and I particularly like this particular study, that try to define a compatibility score in relation to a habitat score, and also taking into account, for instance, farmers' margins, so how much money they can make out of specific crop combinations. And this compatibility score, they're essentially updating it in a database. They're using experimental data, but also say data from other studies and so on, to try to build up some sort of a knowledge base on what crops should we put together. And that could be parsnips with onions, or wheat with maize, or whatever. There's an awful lot of crop species in this particular, that were part of this particular study. But unfortunately, or fortunately or unfortunately, a lot of these spatial, shall we say, optimally spatial combinations are being explored without a genotypic component. Now, I like to think of this as, you can imagine or visualize some sort of a genotypic search space. So you've got, again, I'll talk about parsnips and onions for some reason. You've got your parsnip on the one axis, and you've got your, and what are all these little pixels representing? Well, they're representing essentially a sort of a panel, a core set of genotypes that represent the phenotypic diversity of that species. So this is the parsnip axis, and on here you've got all the other onions that again try to capture the diversity, the phenotypic diversity of the onions on the other axis. And this would, for instance, in this sort of a scenario, is a fairly neutral crop combination. So if it's blue, then it's slightly above average combination. Red might be slightly below average combination, but in general, you can see it's more or less, they're not performing well or badly particularly. You could also have a much better crop combination where a lot more of these pixels are blue, or a very bad combination. And although by chance, you're probably going to land on a fairly representative set of genotypes, there is also the chance that just by you're unlucky and you end up with this particular combination of genotypes that in this case work very badly, and that doesn't really represent the whole potential for performance of those two species. So it's a bit of a conundrum for an experimentalist if you want to test not just which species go well together, but also bring in a genetic component. Well, then the experiments start to explode. So the search space becomes actually too large for us to deal with. If we look here, suppose we're again looking for compatibility between two crops, that's this line here. Once we start introducing a certain number of genotypes, we can see that line already starting to get to enormous numbers of combinations that we need to test. And yeah, heaven forbid we even go to 10 different species. Then the numbers start to become astronomical. And this is even without considering replications. So this is just the number of combinations to begin with that we should start thinking about. So these search spaces are obviously too large. We can't include everything. So one way to try to tackle that is to use a sparse search space, so use an incomplete design, where we accommodate more genotypes of our two species, but instead of testing all possible combinations, we take a subset of them. And they've been shown, again, should work with Benedict and others, to do quite well in getting robust estimates for these genotypes without having to test them against all possible other candidates. But again, I won't go into it, but it's just to say it's a non-trivial problem to explore genotypic search space as well. So it's quite obvious why up to now experiments have only been on one or at most two genotypes, those kind of numbers, when they're doing species intercropping studies. Okay, we can talk more about it if we have time. Now, the final quadrant of this circle is this selection scheme for intercropping. And the first is, of course, the easiest. Select crops separately in monoculture. Very much the simplest approach. And it has, I've motivated this selection efficiency as a way to guide, shall we say, our decision on this. But that's intimately related with just this correlation between performance across systems. And you can see, yeah, this row correlation between, again, if we just rank them in terms of yield production, or we just do a correlation between them, yeah, it's very much expressing the same parameter, shall we say. I'm going to go over that quickly here. Again, I'm running out of time a little bit. So yeah, there has been this nice simulation study that looked at this correlation coefficient. Essentially, it's more or less the same as our selection efficiency. And at higher correlations, well, you get greater genetic gain. So there's, again, more information that's being transmitted or faithfully transmitted between our two systems. And at lower selection efficiencies or at lower correlations, well, then you can see genetic gain is reduced because, again, we're not doing as well in ranking our genotypes in one system for the next. But also the choice of optimal strategy in this simulation actually changed. So it can have influences on what sort of breeding scheme you would set up based on this correlation coefficient. The second point here is, okay, well, suppose we don't have a faithful conservation of performance across systems, but we do know that crops, shall we say, there's a sort of a general mixing ability can be well defined, so that this general mixing effects capture most of the compatibility between our two crops. One way around it then is to, an effective way, shall we say, of using that sort of basically that the choice of specific genotype doesn't matter, is to use what's called testers. And this is very analogous to what happens in hybrid breeding. So in hybrid breeding, it's often in F1 hybrid breeding, you're trying to choose two parents that combine very well to produce a high performance F1 hybrid. So again, do you know what hybrids are? When I say analogous to hybrid breeding, I'm getting some nods here. Yes. Okay, so the idea is you're trying to put together, you're trying to choose two parents that combine well to perform, to produce an F1 hybrid. And in the same way, if we have a sort of a large amount of that, the variation can be captured by by a general mixing effect, well then testers are an efficient way of breeding in this system, or can be. So again, imagine that we've got our two crop species, we have potential candidates, potential genotypes that we would like to combine. We want to see what the best combination is. We could test all possible combinations, and without testers, yeah, we do need per replication n1 times n2 plots, so all possible combinations. We could reduce that number just by using testers, by having a single genotype of the other species. So instead of the product, we just need the sum of those numbers per replicate. And so it's a sort of a halfway house. It's a sort of a compromise. We don't do this simultaneous breeding in the sense of having the full potential. So the set of candidates tested with each other. We simplify the problem, and we would choose these tester lines to be representative of the partner crop. But how do we make that choice? Now, so the final, this sort of the ultimate is to do this co-selection, where again, you're growing these together. You're not necessarily using testers now. You're trying to use some smart way of choosing representative genotypes of the two species. You're growing them together, and you have some framework set up for estimating these direct genetic effects. It allows us to define a selection objective. For instance, if we understand the relative value of these two crops, it's a very simple way of defining a selection index in this case, where we just weight these effects by their economic weight. So essentially the price in euros per kilogram of each of these two species. Okay, yeah, I'll skip over this. Again, I'm going to skip over this. You can have a look at the slides afterwards. Yeah, but if we're dealing with direct and indirect genetic effects, it's quite likely that we have a negative correlation between them, potentially. Certainly for biomass, we've seen this. And why would we have a negative correlation between direct and indirect genetic effects for biomass? Is that obvious for people? Competition, exactly. Yeah, so big neighbors with a big DGE. So a DGE here would be an effect. So it would be the difference between the average, the overall mean, and the performance of that individual. So in this case, big neighbors, so bigger than average neighbors, they're competing more for resources, for nutrients, water, and so on. And similarly, a small neighbor, just less than average, below average size, lower biomass, would be quite the opposite. They probably would be undercompeted. So this competition, indeed, for resources tends to drive a negative correlation between these effects. But what about other traits? Do we always have a negative correlation? Can we think of examples of positive correlations? Yeah.
Plant height could be an obvious trait. A taller plant is more shade to its neighbor, so the neighbor starts to stretch.
Yeah.
The shade avoidance response, so it also becomes taller.
Yes.
That would be potentially positive.
Yes, exactly. So height would be a good example of where you could have tall neighbors force or causing their neighbors to also start to shoot up, and so you get you have taller neighbors ultimately leading to slightly taller plants next to each other and so on. So there you could have a positive a positive correlation. Root proliferation was another point that I thought about. If you had a plant that tends to produce a lot of roots, now its neighbor could just give up and say, okay, I'm not going to proliferate, but oftentimes plants respond by proliferating also in their roots to try to compete for below. So if you were measuring root proliferation, a variety or a genotype that's producing a lot of roots could invoke that response in other neighbors as well.
Can you also look at nutrient sufficiency, or is that too broad? So that should you then look at the plant characteristic like that?
It could be, again, these DGs or IGs could be defined on any trait that you're interested in. So if you have a way of defining, yeah, exactly. If we look at biomass production, again, Benedict showed that there was a negative correlation between, this is yield, sorry, in these mixtures of pea and barley. In Arabidopsis, what I showed earlier, the Arabidopsis study, they found a very tight negative correlation between biomass in these Arabidopsis pots. So again, as expected. But I guess where I want to conclude, now that my time is up, I've got 1 minute. Once we understand the system, so if we're able to measure these parameters, these system parameters, so these variances of our direct and indirect genetic effects, the covariances between them, if we also understand a bit about the crops, one approach will be to simulate a breeding strategy to figure out what would be the best way to improve this system. And this has already been done in the literature. We had a student a number of years ago who did a nice simulation study looking at an intercrop between faba bean and maize. He looked at two scenarios, one where the two crops were equally valuable, and he also considered the more realistic scenario where the faba bean is more valuable than maize. And he set up, he obviously had to choose various different parameters regarding the variances. He also had a negative covariance, shall we say, between the direct and indirect genetic effects. And what he found was, again, it was understandable what we found. But then in the situation where the fallow bean is more valuable than the maize, what happens is that your system starts to diverge towards selecting genotypes of fallow bean that have a large direct genetic effect, and genotypes of maize in green here that have a large indirect genetic effect. So the maize in this system, with these particular economic weightings, the maize starts to become more and more a helper species to promote production of fallow beans. And that comes at the expense of its own performance. Whereas if we have equal weights between these two species, both of these species then start to improve regarding, again, this is the genetic merit, the total genetic value for these effects. But similarly, at the same time, you also start to improve the indirect genetic effects of both species, despite this negative correlation. So essentially, depending on the choice of economic weights, you guide the system regarding how it evolves over multiple rounds and multiple generations of selection. Okay, so that gives us some guiding principles just to conclude with. So if we have quite a high correlation between our monocrop and intercrop, this is this high selection efficiency as well, we can certainly make progress via selection in the monocrop, so breeders don't need to do anything new. But that doesn't give us any information on these IGEs. We cannot learn anything about IGEs by monocrops for an intercropping, because we need to see how this, what's the behavior of the partner species in response to that crop. If we understand about the size of these variances, and particularly the ratio of these variances is important. And at early stages, I imagine that maybe this component is quite small. I imagine that breeders might not initially worry too much about it. But that's not to say in the future, if selection isn't being done, that we cannot start to tap into indirect genetic effects as well for breeding. But that's still a little bit, shall we say, for the future. We're not there yet. And yeah, I think very small point, but you can have these negative correlations between these effects, but as long as it's not absolutely negative, so if there's, in this case, minus 0.55, you can still simultaneously improve both crops. Yeah, that's and also improve on the direct and indirect genetic effects. And these weights, in this case, economic weights, they guide how this direct and indirect genetic effects evolve in the system. There are some references there.